Hello everyone, welcome back to my YouTube channel, The Zone of Anatomy. Today we are going to discuss diabetes, part 2 of diabetes. We have already discussed what is diabetes and the type of diabetes in our previous lecture. The link for the same I have already mentioned in the description box, so you might see it before seeing this lecture. Now, in this lecture, we are going to study about diabetes, ketoacidosis, and the carbohydrate metabolism, in which we are basically focused on how the hormones influence the carbohydrate metabolism. So let's proceed. When we come to a diabetic ketoacidosis, the main characteristic or the definition which we give is that there is an increased in the blood glucose or the blood sugar level, and the another definition which you might see in some books is that it is due to increased in the high levels of blood acid. Blood acids are ketones. So either due to a blood sugar level or due to the high blood acids or the ketones level. As we have already discussed, the diabetic ketoacidosis is the main result of our type 1 diabetes, that is diabetes mellitus. Coming to its characteristic, that how we define what is the diabetic ketoacidosis is actually is. First is there is the lack of insulin. That is a main factor for this might come in the MCQ, so do not forget this. Then there is a blood glucose level high. When we say the person is diabetic, when he or she is having their blood glucose level approx or above 125 mg per dl. So in this case, there is a high blood glucose level and the patient is having high or more than 125 mg per dl blood glucose level. The pH is less, that is 7.3. The serum bicarbonates level is 18 milliequivalent per liter, and the elevated serum ketone levels, that it is also the main factor. That is why we put it in our definition also here. And the dehydration. Why dehydration? Because there is the dryness of mouth, dark circles under the eye, and sore throat. It leads to a dehydration. Coming to our occurrence, at what age we see this type of complicated diabetes or a serious diabetes is at the age younger to 30 years of old, or we say the age up to 30 years of age. And the minority, the patient which we see in this condition less, is approximately 70 years of age. Coming to its fatality rate, since it is a very complicated part, when we do not take insulin doses properly, or we do not pay attention to the diabetes, we take it very easy way, then this condition occurs, and therefore the fatality rate is normally low, that is 1 to 4% or even less than this. So it is a complicated disease, it is a dangerous disease, but the condition is when the patient ignore this condition, or the patient do not pay attention towards its insulin level, or he or she do not go to the doctor for a proper or a regular checkup. Now, it is commonly seen in the patient with diabetes mellitus. We have already discussed it. Coming to its symptom is very easy. Here in a diagram, we have seen many emojis depicting all its symptoms. So, let's discuss what are the symptoms is. First is polyuria. What is polyuria? Polyuria is urination, increase in urination. Here you say that the urination is large, and where the most prominent time or the maximum it attains is at a time during night. Coming to polydipsia. Polydipsia is urge of or excessive thirst. So this is the major condition which we see. Then coming to our weight loss. Weight loss is some or the other way is nice to enhance our physique, but sometimes this excess weight loss also creates a problem like diabetes ketoacidosis. So there is the loss of weight, then we have a fatigue, then we have a dyspepsia, What is dyspnea? That is deep, heavy breathing or the suffocation in the breathing. Then we have a vomiting. Then we have a febrile illness and abdominal pain, which leads to a nausea and the polyphagia. That is excess of to eat. And the another condition which is important, that there is the breathing is giving us a fruity smelling. Then we come to the measurements or the test which we prescribe to the patient is measurement of A1C for diabetes. It is very important for the hemoglobin test. Then blood urea nitrogen, creatinine, serum glucose, electrolytes, pH and serum ketones. It is important to know the condition, complete blood count, and the urinalysis. The treatment which we give is intravenous injections of insulin and the fluids, and monitor our electrolytes and glucose level every time. Coming to the carbohydrate metabolism, so we see carbohydrate metabolism into two phases. First is fed state or absorptive state, or the condition or the time when we eat something, and the time in the fast state when we do overnight fasting. In that case, we see how insulin and glucagon works. So here is a small chart which describes when we use the glucagon and when we use the insulin. When there is hyperglycemic condition, what is hyperglycemic condition? When our blood glucose level increases, and this condition came during our feeding stage or the time when we take the food. In this, we use our insulin. Insulin is released by our beta cells. Coming to your hypoglycemic condition is due to our low blood glucose level, and the hormones which is responsible or which comes into play is our alpha cells, and both these are released from the pancreas, that is endocrine gland, and that is heterocrine gland, type of heterocline or a mixed gland. Coming to the processes is that insulin, how the insulin works. As I say, in the feeding state, we see the play of this insulin. When the person take the food intake, the glucose is converted into the pyruvate. The process is known as the glycolysis. Then it converts to acetyl CoA. Then Krebs cycle comes into play. Then it goes into the mitochondria, ATC works, and it releases the ATP. And what is the role of ATP? So when we talk about the ATP normally makes our cells or the cell membrane electropositive. That is, potassium ions accumulate in this cell membrane during this process. Coming to a second process which is taking in our cell during this feeding state is that we have the mRNA, right? And we know that the mRNA is usually sent this to the rough ER, rough endoplasmic reticulum, where the ribosomes comes and attach to it. Then it sends this to the Golgi body, and Golgi body do all the packaging and then release the protein, that is an insulin and the C peptide. Insulin has basically a two part, that is one is insulin part and one is C peptide part. So it is complete and a insulin molecule and not the different part. It is like same thing. One another protein or one another thing which is released is also amylin. So amylin, insulin, and C peptide are released from the Golgi body during this process. And then when during this process, as we say, initially we have the high glucose level. If we have a high glucose level and it goes into our bloodstream, then we need to check, we need to balance it. And for the correction, what we need to do is we need to lower or we bring our blood glucose level normally, and the hormone which is responsible is insulin. So now the insulin which we had made by the Golgi body, it released from that cells and come to the bloodstream, and through the bloodstream it went into the liver. In the liver, what is the role of glucose? The glucose is converted, the glucose is breakdown in by the process of glycolysis, and by the process of glycogenesis, like the glucose is converted to the glycogen. So what is the, and again, what is the role of insulin in this, in the liver, is it increases the activity of glycolysis and glycogenesis. Okay, coming to the adipose tissue. So in the adipose tissue, normally the glucose is converted to the triglycerides. So how now we see how this glucose is converted to the triglycerides. So what is this? Glucose is converted to dihydroxyacetone phosphatase, and that is ultimately converted into the glycol. Remember that triglyceride consists of two parts. One is glycol and one is fatty acid. So how these two are made, we are seeing in this process. Second, we have glyceraldehyde 3-phosphate. Glucose is converted into glyceraldehyde 3-phosphate, and then the complete glycolysis is occur. Then we have acetyl CoA, and ultimately leads to the fatty acid. So in this procedure, we see how glycol and fatty acids are produced, and eventually these two forms are triglycerides. So we say in the adipose tissue, glucose is converted into triglyceraldehyde by the increase in the uptake of glucose, and this complete process is known as the lipogenesis. So in the insulin, when we talk about the insulin, that is, we say there is the process known as the lipogenesis occurs. That is the synthesis of fatty acids. And when we talk about the glucagon, there you say there is the lipolysis, that is breakdown of the fatty acid. So this is the differences. When we come to the muscles, what is the role of insulin in muscles? That is, since we already know that there is high blood glucose level in the bloodstream, so there is a glucose uptake by the muscles. Then there are also some amino acids goes to the muscles. In muscles, we see glycolysis, that is conversion of glucose into the energy, and we see glycogenesis. What is glycogenesis? That is conversion of glucose to the glycogen, and we have a protein synthesis. Why we have a protein synthesis? Because here there is an amino acid uptake which leads to a protein synthesis. So the insulin affects on liver by increasing glycolysis and glycogenesis. The glucose in adipose tissue is by the formation of triglycerides, glyceraldehyde, that is lipogenesis, and in muscles we see again glycolysis and glycogenesis. Coming to our in the fed state or absorptive state, whatever we have already seen in the previous slide, I have tried to put in the tabular form, and that is it is occur after the meal when we the digestion of carbohydrates, and when the where this digestion of carbohydrates occur in the mouth. So the fed state lasts for about 4 hours. In the 4 hours, this state started. There is a glucose concentration in the bloodstream rises, that you stimulates our pancreatic beta cells to release insulin into the bloodstream. We see in the process 1. Then this initiates the absorption of blood glucose level. We are talking here about how this glucose is then absorbed by the liver, adipose, and the muscle cells. And what are their action? We have seen in the previous slide. Only then inside these cells, glucose is converted to glucose 6-phosphates. Okay, because there is a glucose intake, and then the glycolysis cycle is going to occur in like all the 3 cells. That is liver cells, adipose cells, adipose tissue, and the muscle cells. And the condition in this is that the glucose level is high inside the cell in the fed state, and in this, if the glucose or the if the insulin is deficient, the condition which we defined is diabetes ketoacidosis, the glucose levels remains high. Why? Because there is deficiency of the insulin. So there the insulin normally do not trigger the processes like glycolysis and the glycogenesis. Therefore, the blood glucose level remains high in the patient having a diabetic ketoacidosis. Coming to this is the picturized from that how insulin works. So as I say that the digested nutrients enters the bloodstream. In the bloodstream, when there is a high blood glucose level, the pancreas triggered and it releases the insulin. Then in the bloodstream, we get our insulin also, and we have high blood glucose also. And this release of insulin high blood glucose level goes into three type of cells, that is liver cells, muscle cells, and adipose cells. In the muscle cells, what is its activity? It normally stimulates the like insulin stimulates the glucose like glycolysis and glycogenesis in the muscle cells. What its function? It again increase the uptake of amino acids, which leads to the protein synthesis. Amino acid, which is used to the protein synthesis, and another is glycolysis, which we have already seen that, and the glycogenesis, that is conversion of glucose to the glycogen for the purpose of our energy. Coming to the adipose cells, as we have already see there is the lipogenesis, that is formation of excessive lipids, or the uh, formation of fats. Coming to the effects of insulin, here I have made the summarized form of insulin, whatever we have learned by picturization or by diagrammatic view or by the in previous flow charts. First, the effects of insulin in the carbohydrate metabolism, that is insulin work when there is a like hyperglycemic condition, and the role of insulin is making the condition hypoglycemic, like it needs to decrease our blood glucose level. Then there is decreased blood glucose level, that is our hypoglycemic action. Then it promotes glycolysis and glycogenesis, that is formation of our glucagon and increase our ATP. Then it inhibits gluconeogenesis, that is formation of glucose, and the glycogenolysis, that is breakdown of glycogen to the glucose, because we do not need more glucose when there is a feeding state and when the insulin is so we need to inhibit these two processes over here. And lastly and finally, that the in carbohydrate metabolism, there is increased in insulin dependent transporters, which we normally see on the cells. We term it as a blood transporters. Coming to the protein metabolism, we say it enhances the protein synthesis. How it enhances? By the processes of lipogenesis, as it stimulates the entry of amino acid. Then we come to the effects of fat metabolism, how it effects. First is lipolysis and ketogenesis. Insulin regulates release of free fatty acid from hydrolyzed TGS, triglycerides, which we see in our adipose tissue. Then insulin also decrease the degradation of triglycerides, which will ultimately release the fatty acids. Coming to the condition when the patient is having diabetic ketoacidosis, then the triglyceride stored normally which is stored in our adipose tissue undergo excessive degradation, and that leads to a free fatty acids, and which are converted to the ketone bodies, and excessive ketone bodies it ultimately leads to diabetic ketoacidosis. So here when we discuss in the first slide that the diabetic ketoacidosis definition, that in the diabetic ketoacidosis there might be the high concentration of ketone bodies in the bloodstream. So here is the reason why it happened, and that is due to a simply lipolysis and ketogenesis. Effects of insulin on fat metabolism is lipogenesis. How lipogenesis? That the insulin triggers the effect of triglyceride triglycerides, and that leads to our process of lipogenesis and adipose tissues, that is, insulin stimulates triglyceride synthesis in adipose tissue, which we have already discussed, by enhancing the blood transporters, which we see, and the insulin stimulates fatty acid synthesis by the activation of acetyl CoA. Okay, so we have already seen these two things broadly. And when we come to the insulin stimulate fatty acid, just remember one thing, that this fatty acid and the glycerol makes our triglycerides. So here there is the triglyceride synthesis by enhancing or increasing our glucose transporters, and here is a fatty acid. These fatty acid and glycol mix or form together are triglycerides. Then there is a degradation of chylomicrons or VLDL. Now we see how this helps in our degradation, like how it helps in our condition leading to diabetic ketoacidosis. So coming to it, when we talk about degradation of VLDL cholesterols and the chylomicrons, the insulin stimulates activity of lipoprotein lipase, which is an enzyme. This thing we see in our protein metabolism broadly, then what are chylomicrons, what are VLDL. So in the point which we need to remember in this part of diabetic ketoacidosis is only that VLDL and chylomicrons cannot metabolize at normal rate. Why? Due to the low activity of lipoprotein lipase, that is an enzyme, and that leads to a hypertriglyceridemia. So this insulin leads to the condition hypertriglyceridemia in diabetic ketoacidosis. In carbohydrate metabolism, we say how insulin works. We see the effects of insulin on fat metabolism. So there are two effects which we see, that is lipolysis and ketogenesis, and second is lipogenesis, and third and final we see that whenever someone asks that what are the condition you see, in what type of condition you see in the fat metabolism, when we talk in reference to the diabetic ketoacidosis, just mention one line that in the condition diabetic ketoacidosis, when we talk about the fat metabolism, the condition is known as hypertriglyceridemia, that is increased in the levels of triglyceride height. That's the only thing which we need to remember for carbohydrate metabolism. We need to promote glycolysis and glycogenesis. For fat metabolism, we talk about lipolysis, ketogenesis, and lipogenesis. And ultimately, hypertriglyceridemia leads to a condition for which we are talking since long, that is diabetic ketoacidosis. Coming to our another slide, that is glucagon, which we already see that it works in the fasting state. So the process is similar. There is a decreased glucose entry cell because we are in a fasting state. We are not eating anything. Then it comes, whatever glucose it is making, whatever glucose present in our body is converted to pyruvate, then acetyl CoA, Krebs cycle, etc. that releases ATP, and this ATP depolarizes our membrane, and the glucagon, this condition ultimately leads to our glucagon releases into the bloodstream, and this glucagon then ultimately goes to the liver, adipose tissue, and muscle, as we see in the case of the insulin. In insulin also, the insulin released in the bloodstream. So here in the fasting state, we see glucagon released in the liver, adipose tissue, and the muscle. Now, how these, when we talk about fasting state, the first thing which we need to remember is that in this state, we have a low blood glucose level, and the correction which we need to do is we need to elevate, or we need to increase our blood glucose level. So how we can do is, the whatever glucagon is released in the bloodstream, it started enhancing the process called as glycogenolysis, that is breakdown of glycogen into glucose. Because we have a glycogen with our cell. As a result of this increase in bloodstream, or it becomes normal, blood glucose level become normal in the liver by the process known as the glycogenolysis. Coming to our adipose tissue. When we talk about the adipose tissue, then here we have a lipolysis. In the effects of insulin, we see there is lipogenesis. Here is lipolysis, that is triglyceraldehyde is broken down into fatty acids, and gluconeogenesis, that is breakdown of glucose, that is breakdown of glycogen into the, that is breakdown, that is the formation of glucose mainly, or in a fatty acid undergoes to liver, and it is the basically a process how we make a fatty acid, that the whatever fatty acid we are having in our liver, it gets converted to the fatty acid, and that leads to the, that converts into the glucose. In this case, we just need to make glucose. So first, in adipose tissue, we see there is a lipolysis that makes our fatty acid, and the fatty acids makes glucose, and we have a gluconeogenesis, that is formation of glucose from the non-carbohydrate components. When, as we see, there is a lipolysis that occurs in the adipose tissue. We have a triglycerides, and triglycerides is converted into two parts, that is fatty acid and the glycerol. Whereas in insulin, we see that the glycerol and fatty acid makes a triglycerides. So there is the opposition in this. When we come to how this glucose is formed, this fatty acid goes into the liver, and in the liver, this this glycerol and fatty acid goes into the liver, and there it converted back to the glucose, and it increases the congestion, or it increases the glucose and make hyperglycemia. So it is very easy process. The things which we need to remember in glucagon is in liver, there is glycogenolysis, that is formation of glucose, and also there is adipose tissue, that is gluconeogenesis. Again, we are forming the glucose, and lipolysis, again we are forming the glucose. So ultimately in glucagon we need to make extra, or we need to make glucose. Effects of glucagon which we have seen, that is carbohydrate in the like we have divided into three parts. Effects of glucagon on carbohydrate metabolism, how it affects protein metabolism and fat metabolism. Carbohydrate metabolism, hyperglycemic action, we are increasing our blood glucose level. We stimulate the breakdown of glycogen, that is glycogenolysis. We stimulate the synthesis of glucose via glyconeogenesis from the non-carbohydrate parts in our liver. Then we inhibit glycogen synthesis, that is glycogenesis, and glucose is primarily act on the liver. And the point I have mentioned here, MCQ, it is very important point, is that glucagon primarily acts on a liver and does not influence on the muscle. That's on the previous slide we need to study only the effects of glucagon on our liver and on our adipose tissue and not on the muscles. So the examiner might ask you why this glucagon do not act on the, like it only acts on liver and adipose tissue and not on our muscles. It is true or false? So it is true. Coming to the glucagon, it is a potent hyperglycemic hormone. It is the direct MCQ which might come. How the glucagon affects a protein metabolism? It stimulates the uptake of amino acids by the liver, and then this amino acid is converted into our glucose. So here, like in the adipose tissue, we make our through lipolysis, that is breakdown of TGS into the amino acid, or the glycerol and the fatty acid, and then these amino acid goes back in the liver, and there they started their gluconeogenic pathway and converted it into the glucose. In the fat metabolism, how we see that triglycerides stored in the adipose tissue that I've already explained you, and protein metabolism, amino acid uptake increases. So these are the key points which we need to remember whenever they talk about the carbohydrate metabolism or the effects in our fasting state. So here you can give the answer. This is the picturized form of the diagrammatic view of the glucagon, how it works, so that it is a fasting state. So whatever glucose the body is having, it gets pancreas release glucagon and it stimulates to the like that glucagon goes to, like we have very small glucose concentration, like green points you are seeing here, they are very small as compared to the glucagon. So these glucagon goes into the liver cell. There they do processes to make the more and more and more glucose. So in here we see in liver cells it makes, in adipose cells it makes, but in muscle cells it do not work. So in muscle cells we are not talking about the glucagon. So that's all from my side on this topic, that is carbohydrate metabolism and diabetic ketoacidosis. Hope you enjoyed this lecture. Or like these are the key points or the main important points that the examiner asks, and the things which we need to see. Look, we cannot revise each and everything briefly, or we cannot deeply understand everything. So in my slides, whatever I have explained it, these are very precise and very accurate. Like these points you just need to remember, and all the MCQs, all the answers you can give easily, and you can easily correlate diabetic ketoacidosis with the carbohydrate metabolism. That makes your way very easy. So just revise these slides, just go through these slides, and all the best for your exams. So that's all from my side for today. If you like the content of my video, do like, subscribe, share as much as you can so your friends get benefits and they also feel easy to understand the most complicated topic in the biochemistry. And if you like the channel or the content of the channel, do put your reviews in the comment section so it might help others to visit the channel. So guys, thanks for your support. We'll meet in the next lecture. Till then, take care. Bye.